For 8 hours?
Only allowed to broadcast for up to 8 hours, says YouTube. Shucks. Hi, everyone. Can you hear us? I need some affirmations from the chat room. Ooh, Gord sees Blair and Justin. That's a good sign. How are you doing, Justin? You think we can do this without her?
No.
It's we're like, once or twice a year. We're like, oh, yikes.
Even the disclaimer's broken.
Yeah, I know. What is it? And that's not... There's nothing different about that.
I normally do it first.
Yeah.
But I got sidetracked into the stories so heavily that by the time it was showtime, I realized, oh boy, I'm out of time. That's all right.
Out of time.
This will be the shortest.
Out of time. Let me just real quick check and make sure this works. Best sound quality ever. Okay.
Woo!
Okay.
Oh, is there a chat room?
Yeah. Yeah, of course. There's always a chat room. All right. Are you ready? We're going to start. It's 8:02.
Yeah, I'm ready. I'm always ready.
Okay. All right.
You know what you're going to say?
Yeah, I got... Will you... Okay. 3, 2. This is TWIS. This Week in Science, episode number 669, recorded on Wednesday, May 2nd, 2018. Jeepers Creepers. Hi, everyone. I'm Blair Bazdarich. We don't have Dr. Kiki tonight, but we still have lots of science. We are going to fill your head with old spiders, dino birds, and ancient DNA. But first...
Disclaimer, disclaimer, disclaimer. Despite not writing a disclaimer in time for today's episode, something must be said here at the beginning of the show. So for that reason and that reason only, I am just saying This Week in Science coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Blair.
Good science to you, Justin. Welcome, everyone here and out there to this week episodes of This Week in Science. We have an amazing show ahead. Again, we don't have Dr. Kiki tonight, so we're going to do the best we can at bringing the science. I still found lots of cool stuff to talk about. What did you bring, Justin?
I've got a bunch of ancient people's DNA stories. Ooh, first bird, maybe? Proto-bird, pre-bird?
Ooh.
Dinosaur, probably. And, and something else. Ooh, and Stephen Hawking.
Very interesting. Okay. All right. Very neat. I brought, oh my goodness, I brought so many things. But just for a little toe dip into the pool of science that I brought today, I brought some mosquito news, some bat news, some mantis shrimp news, some interesting stuff. So let's, before we jump into the show, let's just remind everyone, you can subscribe to TWIS on iTunes, Google Play, Stitcher, Spreaker, anywhere you find podcasts, you can find us. Just search for This Week in Science. You can also find us on YouTube if you like looking at our beautiful faces, or on Facebook by searching for This Week in Science, or just go to twis.org. So it's time for some science. Justin, what would you like to tell us about?
Oh, am I up first?
You're up first.
Well, it's because normally that is okay. This is University of Kansas anthropological geneticist Michael Crawford and an international research team. They have narrowed down the Native American founding population that came over from Siberia to approximately 250 people. That was it. from 250 to like 40 million.
That's a narrow gene pool.
Yes. Yeah. So the study, How Strong Was the Bottleneck Associated with the Peopling of the Americas? New Insights from Multi-Sequence Data, is now published in the journal Genetics and Molecular Biology. And it's exactly what they say. Going from that first few hundred, they can actually sort of now see it's an incredible opportunity to evaluate the adaptive evolution that took place, because you have 250 people who aren't all that different from each other. So researchers examined 9 non-coding regions of the DNA samples collected from populations to trace this path. This was including populations, individuals in China, 10 Siberian groups, 10 Native American populations that were scattered across the Americas, Central and South America. The Crawford, actually, I guess he was one of the first non-Soviet researchers to go into Siberia back in 1989 after the breakup of the Soviet Union. And he's worked on Aleut and Eskimo migration populations from Siberia this whole time. It says, it's difficult to go back in time and follow the populations, but we can characterize contemporary genetic diversity and estimate the mutation rates that have occurred in different regions. So, yeah. So back in 2015, he was part of a group that found that the ancestors of all present-day Native Americans entered the Americas from Siberia into what is now Alaska, no earlier than 23 years ago. They also found at that time that the group did split into a couple groups and maybe spent no more than about 8,000, but around 8,000 years of isolation in Beringia. So this is this region in Alaska and Yukon, Canada. They were there for about 8,000 years before the giant glaciers on the Americas melted enough for them to spread out to the rest. So they did DNA sequencing that spanned over 15,000 years of samples, along with computer modeling, and it suggests 250-ish people made up the founding population. And so what they can do with this is that you can now look at existing genetics in a very diverse population, diverse regions, all the way through jungles of South America, the deserts of the Southwest United States, all these different regions, and see how perhaps environment or other stressors caused evolution, how adaptation took place in a human model. So pretty exciting data sets that they're working with now.
That's pretty cool. I think that it's also, this is a really good example of how we think about genetic bottlenecks. And I know I, at least a lot of the time, hear that and I go, oh no, we're doomed. But then there's all these other factors, right? So some people thought it was all about genetics and some people thought it was all about adaptation. And it's, of course, it's a little column A, it's a little column B, it's a little epigenetics, a little bit of all this other stuff happening. And so this is a perfect example of how 250 individuals might sound like quite the genetic bottleneck, but there's still opportunity for change in a population and it can blossom to millions of individuals. It's very interesting. And I think that it's an important lesson for that and something that a lot of us can relate to because it's humans.
Yeah. And they're pretty distinct populations too. Mm-hmm. As you head down into South America, or there's a repopulating event too that takes place. So these folks were 8,000 years in this sort of ice age oasis of the ice age up in Beringia, and made it down into the lower states. And then for some reason, some of them were like, it's getting too warm down here. They went back up and crossed over and ended up in Greenland. So the Greenlandic population is largely a sort of dropping down into the Americas and then leaving again.
Yeah. Or maybe there weren't as many elk as they had hoped down there.
well, actually, the story is that that population was led there by somebody who had a vision of right, and so he was fun, right. Backstory to before he had this group that was following him in his vision quest is he may have murdered somebody.
Oh, I
and
see.
Was no longer welcome where he had been living and had to take off.
Attention, everyone. I had a vision that you should ignore anything anyone says about me and follow me right now. Let's go.
Yes,
Pack up. No time. Let's go.
And his vision panned out. He had this vision of these other peoples, and they found other people. There were already Inuits there who hadn't done the drop down crossover. They just sort of come across Canada, and also that was a little bit interesting there because when they got there, you mentioned there weren't as many elk or something to hunt, right?
Yeah.
When they got there, there were muskoxes
on
Greenland.
So much meat on a muskox
that weren't being hunted.
Yeah.
Because they were dirty animals. And the Inuits who were there were using sealskin to stay warm, which was not very effective.
Mm-hmm. When you take all the blubber off, not so warming anymore.
Not so warm. So they're like freezing and hungry. And there's all these like furry muskox that they're like, oh, dirty animal. Wouldn't want to eat it. No, they're just going to fish. So they changed that was sort of I guess a failing population at the time. So,
well, I'll tell you, I'm not one for furs, but if I was going to pick one, muskox would be pretty cool. Quite fluffy. Nice. Excellent. Well,
did you bring any stories, Blair?
I brought a lot, although I just realized they kind of all belong in the animal corner.
That's much to be expected.
So I might just start the corner up now and we'll have an extended version. Sound good?
Sounds good.
What time is it?
We're 13 minutes in. Right.
Oh, okay. So it's time for Blair's Animal Corner. Here we go.
Oh no, that's what you're saying.
Yeah.
Dogs are creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Well, I'm going to start with a story that is about mosquitoes, but is also therefore very much about humans. So mosquitoes, as you probably are aware, the males are not the ones who bite and make us itch. Those are the females. And what a lot of people also don't realize is they are biting us and they are feeding on human blood, not for nutrition, but actually because there's protein and other components, other building blocks in our blood that they use to make eggs.
What?
Yeah, so that's why, or that's until now, dun dun dun, why they thought that mosquitoes bit us and drank our blood was really just to make eggs. But now
it's for sport.
You're not that far off. So the University of Cincinnati did a study that was actually quite accidental in its origination. So they actually found out that they had this batch of thirsty, dehydrated mosquitoes that some of them escaped in the lab. And what they discovered the hard way was that those mosquitoes were unusually aggressive. They were all trying to bite all over everybody in the lab. So they saw something very interesting here. These dehydrated, these very thirsty mosquitoes were being very aggressive. So now this is a good question about what happens when a mosquito is thirsty. Usually we think about mosquitoes and mosquito population dynamics around wet times because you think about standing water. That's what you don't want because that's where they lay those eggs that they made out of your blood. But it turns out that lack of water might also be pretty important. So University of Cincinnati, they then took mosquitoes in their biology lab, which they describe as adults of 6 different species sequestered in large mesh boxes in a room kept at a balmy 82 degrees. Each box has a source of water and nectar and a place to lay eggs to create a constant supply of lab specimens. And they say it is like the Ritz-Carlton in there for mosquitoes. But so in this study, they decided to take some of these mosquitoes and they theorized that dehydration would actually lead to suppressed metabolism, reduced activity, and decreased blood feeding due to just being laid out, pretty much. What they found instead was that dehydration changed them to the opposite. They found that only 5 to 10% of female mosquitoes feeds at any time when they have normal access to water and food. But when you dehydrate them, that prompted as many as 30% of female mosquitoes to seek blood. So what they found was that dehydration increases propensity of mosquitoes to land and feed on hosts. Mosquitoes that had access to water when they were higher in temperature and humidity still did not seek increased blood meals. So it's really just they're thirsty. They're going for blood because they're thirsty. So they release some adult mosquitoes in a large mesh enclosure that mimics conditions in nature. And then they put a membrane-covered disc of blood that mimics a bird or animal host, and then they recorded the occasions a day after rain and a day after a dry spell to see what they saw them doing in the two. And so they really did see that dehydrated mosquitoes were more likely to seek blood. So why is this important?
Because now, if you're having an outdoor event, you can leave out really tiny glasses of water all around the perimeter.
Right. Right. This is actually super important because what are we seeing more often now with climate change? Drought. And so as drought happens, we can now potentially project a change in mosquito behavior. Remembering, of course, that mosquitoes bring a lot of really dangerous things. They have a huge toll on public health because of malaria, because of now Zika also. So if they know that drought is likely to bring more mosquitoes and more mosquito bites, then they can get ahead of these public health crises.
So this is sort of ruining one of my attempts at finding silver linings in the face of global warming, which is prolonged dry spell, which global warming isn't necessarily, it just depends.
It increases the severity and the likelihood of drought in a lot of places.
In a lot of places. Other places you're going to be wetter or just have snow.
So it increases the likelihood of extreme weather events. So if you're a place that has droughts, you're likely to have more droughts.
Right.
If you're a place that does not,
it does. California is drought prone.
Right.
And one of the silver linings I was hoping for was that meant less standing water and a smaller mosquito population. But if those smaller mosquito populations are just going to be a lot more aggressive about biting people, then the whole silver lining thing is gone.
Yeah, absolutely. So this is another situation. Darned if you do, darned if you don't. The mosquitoes are going to cause problems. Now, I will just, as a quick disclaimer, remind everyone that through This Week in Science, we discovered that mosquitoes are also pollinators. So we can't just do away with them completely. But what we can do is anticipate that if droughts are going to make mosquitoes more likely to bite, we can try harder to vaccinate, spread out more mosquito nets, do other things that we can do to reduce the number of instances during which you're bitten.
I wonder if you could leave out like a hummingbird feeder, but like a really small version
with blood in it. Yeah,
blood in it. That's
a great idea.
So that way, you put one outside the window, and then if you got the hole in the screen, you'll be fine. They'll just stop by the little blood drinking fountain and be gone.
Yeah, absolutely. So maybe blood feeders are the wave of the future. Who knows? Speaking of creepy things, I want to talk about the mantis shrimp. So we love the mantis shrimp on this show. The mantis shrimp is amazing for a bunch of reasons, but you can't help but admit that they're kind of crazy looking, kind of creepy looking. So we've talked a lot about the mantis shrimp, their stabby tendencies and their slammy tendencies, but also their spectacular eyeballs. So their eyes are fascinating because they see colors that we're not, that we can't even fathom. They have so many cones. I think they have 12 different kinds of cones instead of the 3 that we have. Their eyes are just insane. But now an extra layer of sight insanity on the mantis shrimp comes from University of Bristol. Looking at their eyes, they say that they are the most mobile eyes in the animal kingdom. So if you ever see a video of a mantis shrimp, that you'll see that their eyes are constantly moving up, down, side to side, and also rotating on their axes.
And they're on like a stem thing. They're like
yes,
implanted in there, embedded in there.
Yes. And they're independent of each other. So they can move in pitch, that's up and down. Yaw is side to side, and they can roll. They can twist around the eye stalk, and they can do this independently from one another. So what University of Bristol wanted to see was why it seems like they never stop moving, how they avoid blurring their vision. Because if you imagine you're doing this all the time, you'd have trouble focusing on things. And why are they moving it so much? So what they did is they wanted to see what this this had anything to do at all with stabilization. So they found that mantis shrimp, they make stabilizing side-to-side movements that help keep their vision steady as they move. But then when they dug deeper, they found that while they are stabilizing in the horizontal direction, they're still rolling their eyes. If you imagine that you're like adjusting binoculars, that's what I'm doing if you're just listening. But that's kind of how they're rolling their eyes around this central axis, even when they're stabilizing them left to right. So what they then did is they wanted to see what would happen if they put them in something that was moving them around. So if kind of like a giant, like one of those county fair structures maybe that you've walked through where it's a big tube that's kind of turning. I'm so dating myself right now. Who goes to the county fair anymore? But you would walk through this thing and it'd kind of be moving and you'd have to adjust your body like this. So they had a thing that did that on all different dimensions. So they were moving in space and they were trying to keep their eyes on a fixed object. And they found that actually didn't have a lot of effect on the shrimps at all. No matter what position their eyes moved in or their body moved in, they can still reliably and accurately follow the motion of a pattern that's moving sideways. Yeah. So as they did this, they were trying to essentially give the mantis shrimp vertigo. And they found that in response to things moving around them, the mantis shrimp did not roll their eyes in any conjunction with their surroundings moving around them. So the mantis shrimp is completely immune from the effects of rolling eyes and changing visual planes around them. So this stuff has no effect on their space, on their eyes, on the rolling of their eyes. This is unprecedented and just confusing. So even when their eyes are completely sideways, they can see fine.
But of course, none of us see with our eyes.
Right.
I mean, yes. We see with the brain. And so if their brain is perfectly used to accepting this information, I'm sure it's making plenty of sense out of it. And it kind of already makes me think of like, well, like I'm used to having sort of binocular predator vision, right? Whereas like a lot of prey species have their eyes way on the sides of their heads so they can see a 360. That would be weird to us if suddenly we were looking both left and right at the same time. That would be a really odd feeling.
Agree.
Right? And our brains would have a hard time processing that. Another one that comes to mind is eagles. I think eagles do a sort of rapid eye movement scanning thing when they're flying up above a field. And what that sort of allows them to do is pick up motion a little bit better. Because even though they're moving their eyes rapidly, when there is a little when they settle on something, when they focus on something, that vision is a little bit more crisp than it was if they had just been staring at something for a while. And it may also assist them by their way their brain works in picking up motion from a distance. So
yeah, absolutely. And so we've before on the show talked about how recent research looking at all these different potential colors that the mantis shrimp can see might actually be a reflection of the fact that they can't see much. And so because their vision is so bad, they have to compensate with extra cones. So this could actually be something where they might not be able to see that well at all. They're just seeing kind of blurry clumps, and they don't need to be able to see good enough so that moving their eyes around like this doesn't actually affect their picture much at all. If it's low def, it doesn't really matter. But what is really the question then, and what has got these researchers really puzzled and excited for their next study, is if that's the case, if this doesn't affect them, all of this eye movement, why are they rolling their eyes at all?
Because they're just massively unimpressed with everything.
Yeah. They're just constantly there they go. Oh, there's that blobfish again. Oh my God. Stop complaining, blobfish.
No, it's passive aggressive.
Yes. But why is this happening? Why are they moving their eyes constantly if they don't adjust that behavior in relation to space moving around them? It's maybe they're not eyes at all.
Or
maybe they're blind. Maybe they can't see at all. Who knows?
Well, I figure this is something that gets eaten by other things. So there's
sometimes they're pretty good at defending themselves, but they can be eaten by other things.
So there's that sort of keeping an eye out in every direction for stuff going on. But maybe it's not something they have any sort of control over, right?
Mm-hmm.
Maybe their eyes are just sort of like a heartbeat or a breathing pattern, and
or they're just constantly being moved by the ocean.
Maybe they're just the same buoyancy, and they're just
whoa, yeah. Well, who knows? We'll find out, hopefully. But the mantis shrimp is just an enigma. Speaking of vision and predator vision, actually, I have the latest news in camouflage, which is a thing that I've known about since I was very small, but I've learned as the years progress, there's more to know about camouflage. It's not just about blending in with your surroundings. It's so much more complicated and specialized than that. And we've talked a bunch on the show about really weird kinds of camouflage, like zebra stripes, and potential reasons for them being the way that they are. But the type of camouflage that I find the most interesting and often the most beautiful is called disruptive coloration, which we've talked about on the show before. And this is basically just a type of camouflage that breaks up the body shape of an animal, so that even if they're not camouflaged, it is hard to recognize what the shape of that animal is. This is something that has been theorized, but has not been confirmed until today. So, University of Stirling and Abertay University, both in Scotland, I had to look them up. They looked at a particular camouflage found in pythons, moths, and frogs. Say that 5 times fast. Pythons, moths, and frogs. And they found that they have something very special called edge-enhanced disruptive coloration. So this means there are dark patches of camouflage on the edges. And then they have so half of them has darker edges and half of them has lighter edges. And so this makes them difficult to identify as a particular type of thing. So the idea is this gives the impression of shadows and depth, and it slows the process of recognition. In the past, says Dr. Sharman in the study, it has been assumed that if you can detect where a target is, you must also know what it is. But for the first time, we show this is not the case. So in this study, they had human volunteers. So here's also asterisk, 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 human volunteers. Our vision, as just discussed, not the best in the animal kingdom. We have this binocular vision. We are predators, but we're kind of lazy and out of practice. So I would say we need to test this on some really hardcore predators to know for sure that this was working to the effect that they claim. But what they did is they took these human volunteers, they had them view images of camouflaged animals on a monitor, and then they were asked to locate the target and identify it as either predator or prey. They found that participants required longer to identify a target, even if they located it quickly. So it took them a longer time to figure out what it was. And then even when they simplified the background, so they kind of did this like pebbly, here, let me see if I can screen share for you. So they did this pebbly background. The pebbly background was supposed to help the shape of a bunny rabbit, let's say, blend in. And they had these different kinds.
One of those charts that they use to mess with colorblind people.
Yeah, absolutely. So I would have failed this test for sure. But so you can see, this is so hard for me. But so this is an animal that has edge-enhanced disruptive coloration. So certain edges are different colors. Then you have an animal that has a uniform color match. So they found a color in the palette behind them, and they just colored it all that color. And then they have flat disruptive coloration in the middle. So that's coloration where they just have the fancy patterning, but they don't have the edge enhanced element. And so when they did that, they found that if you had edge enhanced disruptive coloration, they had a lot of trouble finding, and they had more trouble identifying them after they found them. So this gives a prey animal an extra beat, an extra few seconds to escape capture after they've been discovered. So, as I said, this is something that we've been theorizing for decades. This is something that I learned about in college, but this is something that we didn't have evidence for. This is just something that was observed and assumed, and we were right, but now it's actually been proven, at least with human volunteers at this point. So the other kind of asterisk to this is that this does potentially have some pretty interesting applications, of course, in the use of camouflage in military context, which I was very surprised to hear that the current military camouflage that's used is not developed on empirical evidence. So there's no research saying that any particular type of camouflage is best for military use. So this is actually something that could affect some real world applications. Pretty interesting. Yeah. There you go. Camouflage. We're still learning new things about it. All right. I think it might be time for a break. Look at that.
Oh, yeah.
I think we are going to take a quick break, and then we're going to have more science for you. Stay tuned for more This Week in Science. Did you know that TWIS has merchandise you might enjoy?
What?
Oh, yes. So Justin's helping me out with the break. This is fun. So if you head on over to TWIS.org. So let's see. Where's my... Ooh, I'm skipping ahead. Here's TWIS.org. There's all sorts of fun stuff on our website. But in particular, if you click on this link that says Zazzle store, you'll be transported to our Zazzle store.
How come it looks like an art supply store?
I don't know, man. But there's our logo.
We never got around to changing the default.
I don't know, man. It's fine. I used those things when I made the things.
Okay.
So if you go to TWIS.org and then you click on the Zazzle tab, you'll find all sorts of cool stuff. So just at first, you'll see the most popular things, the things that have been purchased the most. We have mouse pads, we have mugs, we have t-shirts, we have pillows, we have stamps. Oh, I need to buy those stamps. We have hats that we've signed at live shows. Those are always fun. And then we have mugs, and we have all sorts of cool stuff. And if you go to all of the products, if you just click on the products tab, you can scroll through so many things. Ooh, look, an apron. So this is something that is a really great place to go for the TWIS lover in your life, for the science lover. And so this is a great way to support TWIS as well. Percentage of the proceeds from that do go to support the show. And as an added bonus, if you have any particular images from previous calendars that you want on any particular Zazzle products, if you send me a message either through Facebook or Twitter or email, more on that later, you can actually request for certain items, and I am likely to be able to put them into the store. So we've already done that a couple times with the TWIS. The polo here. We have a couple high school teachers that listen to TWIS, and they wanted a TWIS polo because they had a what's it called, a dress code at their school. And we put that up there, and some people have bought them to wear in their science classrooms, which is so cool. So go ahead and check that out. You can also help us just by donating. Actually, TWIS is mainly supported by listeners like you.
Wait, so there's another way that we're supported?
Yes. So it's not just by buying stuff, because, not everyone's into stuff. Maybe you already have all of your TWIS stuff. You can also just support the show. And those donations, they go to a lot of things. They go to our hosting, our bandwidth, contractors we need to hire, fun things we do for the show, when we travel for live shows, all that kind of good stuff. We appreciate any amount of donation, $1, $2, $5, $10, $100 if you have it.
We probably appreciate the larger numbers more per.
Justin, I don't know about you, but I value all of our donors equally.
I didn't say that I wouldn't value the other ones, but I say in a denominational sense, you would have to.
I mean, people know to give us what they can. I think that goes without saying. We could use whatever you have to give. And we can take that in a couple of ways. So if you go to twis.org, you'll find PayPal buttons all over the website. It's just all over. So on the front page,
actually the entire website. You can click
click anywhere. No, that's not actually true. There's PayPal buttons. They're very obvious. They're on the show pages. You can make a straight donation to us, and then all the money, minus a couple of fees, goes straight to us. The other way that you can support is through Patreon. And Patreon is kind of like Kickstarter for producers of media or content. And you can find us at patreon.com/thisweekinscience. Depending on your donation level, you get different sorts of kickbacks. So, for example, I have not sent out an original Blair's Animal Corner piece of art in a long time because we don't have any of the donors at that level right now. But if you donate a certain amount per month, I think it's $15 a month, I don't remember, but if you donate a certain amount, you can get an original piece of art from me. You also can get shout outs on the show. You can do all sorts of amazing things. You can get all sorts of cool stuff, t-shirts, by helping support us on Patreon.
And the best way to do it with Patreon is that with Patreon, right, you can set up a reoccurring gift. And
you can do that on PayPal too, actually.
Oh, so wow. Those are the best kinds, because then even if you're not thinking about us, you totally forgot about us,
then we're still thinking about you.
You'll still be sending us your donation every month until you probably change cards, and it just by default then doesn't allow it to go through. Right. So that's the one we like the most.
Absolutely. And if you can't afford a donation, everybody who listens to this show can still help us. And you can do that by spreading the word. So tell people about the show. Get people to listen. Send them a link to a particular show you think they might enjoy. Use your social networks, any social medias you enjoy. Let people know about This Week in Science so that we can spread the science far and wide.
And even if you have donated, you can still do all those things.
That's also true. And the other thing that you can do that's really important is you can give us a review on iTunes. So,
but it has to be a good one.
it would be helpful if it were a good one, but we would like you to review us, because you also then rating the reviews that you think are helpful helps, because they push the most quote unquote relevant reviews to the top. We have a review from like 4 years ago at the top of our iTunes reviews right now, and that's not super helpful to people who are trying to find us. So give us a review, upvote reviews that you think are helpful, but also just thank you for being here, and thank you for being a part of the TWIS Science community, because we would not and we could not do it without you. Thanks so much.
And we're back with more This Week in Science.
Justin, do you know what time it is now?
I do. I just don't know whose segment it's going to be.
I'll do it because I found the message. I did all of the fact finding. So right now it is time for What Has Science Done For Me Lately? That's right. So tonight we have a message from Carin in Perth in Western Australia. Carin says, "Hi, TWIS gang. I am a relatively new listener. I've only been listening to the podcast for a few months, but I love listening and I really appreciate the variety of stories that you share. As a biology nut, it's great hearing stories from outside my usual field. I really enjoy listening to the What Has Science Done For Me Lately segment, and it always gets me thinking, what would I share? I thought about the medical advancements that have helped me or my family and friends, but don't have any spectacular stories like others have had. Then I thought about the fact that I work with animals and the constant research that improves my ability to look after my animals and even look after animals in the wild. Then I thought about the technological advancements helping us to protect the environment and maybe even mitigate climate change and other issues. But none of these things seemed enough. None of them really shares what science does for me and why I love science. Then I realized I love science. I love learning new things, understanding new things, and everyday things. I love the huh and wow moments you get in science. I love that with science, we will never stop learning. What has science done for me lately? Every day, science makes me happy. And I think that should count for something."
It does.
That's awesome. Thank you so much, Carin. I loved that. That was straight out of my heart and your heart connected here through the internet and TWIS. So remember, everyone, we need you to write in to let us know what science has done for you lately. What does it do for you every day? This segment is so important to us in all times, but especially now. We want to help remind everyone why science is important and why it's important to talk about. So if you would like to share what science has done for you lately, please leave a message on our Facebook page. That's at facebook.com/thisweekinscience. Or you can email Dr. Kiki directly at, it's Kirsten at thisweekinscience.com. Sorry, it was wrong in the notes. Kirsten at thisweekinscience.com. We want to fill this segment every week, forever. So keep sending those stories our way. We love hearing them. They really warm our heart and remind us why we're here. Isn't that right, Justin?
It's true. It's very true.
It's true. All right, Justin, time for more science. What do you got?
Let's see. Okay, let's go to, if you want to learn a bit about your ancestral roots, you can spit a little bit of saliva into an Eppendorf tube, send it off to a sequencing company to analyze your DNA. But if you've been dead for 1,000 years, you are likely out of spit, making it much more difficult to get your results. Unless you planned ahead, which is exactly what some forward-thinking people of the Mule Spring Rockshelter did. While the discovery of DNA sequencing technology and Eppendorf tubes were still several hundred years away, folks in the foothills of the Spring Mountains of southern Nevada gathered agave and yucca plants for food. The artichoke-like hearts and inner leaves of the plants were roasted, then chewed to consume the sweet pulp. And once the gum lost its flavor, they did what many gum chewers still do to this day. They simply spat it out wherever they were. This left wads of stringy fibers that they called quids. In the late 60s, researchers from Nevada's Desert Research Institute and the University of Nevada, Las Vegas recovered thousands of these curious quids. They put them into storage without any consideration for DNA preservation because that still wasn't a thing. Now, half a century later, a research team decided to re-examine the quid specimens and see if they could extract DNA from them. This is quotey voice of Susan Edwards, who's an associate research archaeologist at the Desert Research Institute, one of the co-principal investigators who came up with the idea of doing this. The quid's coarse texture is excellent for capturing skin cells from the mouth, making them the equivalent of a modern-day cheek swab. We were surprised, says Dwayne Moser, PhD associate research professor of microbiology. We were surprised by the consistency with which we were able to recover intact human DNA from this common type of plant-based artifact. Since these materials were also radiocarbon dated, in essence, they provide a time-resolved hotel registry for this unique site over a period of hundreds of years. Research on this is published in the public library, Science Online. So that's really cool. So even though they sort of missed the, just order one of those DNA kits and then spit into the thing, they still got sequenced.
Oh,
and
that's
what's really brilliant is they didn't even have to pay. They got it for free.
That's awesome. That's wow. Gum. Silly gum. Quids.
Quids. I got another question for you, though.
Yeah.
How far back in history do you need to go to find a bird that's no longer a bird? Or if you go the other way, how far removed from a dinosaur, or how dinosaur-like, or how bird-like, the differentiation before we start going, that's bird.
Well, let's see. Archaeopteryx was 150 million years ago.
Has feathers. Has wings.
Has little teethers, though.
Has teeth. So, can a bird have teeth?
I don't know. You tell me.
How about a beak? Does it have to have a beak?
Well, I would say that's a good question. I would say so. I think that birds have calcified eggs, right? They have feathers. No?
Oh no, okay.
Yeah. Okay, so they have calcified eggs. They have feathers.
Are dinosaur eggs calcified?
I think so.
Dinosaur eggs.
Okay. Yeah. Okay. They have hollow bones.
Ooh, the hollow bone one's a good one.
And they're warm-blooded. Those are birds. So Archaeopteryx, I don't know if Archaeopteryx had all of those things. That's a good question. I'm not well versed. So what did you find?
So this is 100 million years ago in North America. There was something that looked like a toothy seagull.
Okay.
Ichthyornis dispar. It drew the attention of some famous naturalists. O.C. Marsh out of Yale, who was the first person to describe it. Charles Darwin was also very fascinated with this creature. Yet despite the existence of partial specimens, there had been no significant new skull material beyond the fragmentary remains found back in the 1870s. Now, a Yale-led team reports on new specimens with three-dimensional cranial remains, including one example of a complete skull and two previously overlooked cranial elements that were in the original specimen, revealing new details about these striking transformation in evolutionary history. So this is 100 million years ago. So this is still firmly in the age of the dinosaur, giant lizardy times. But this is, they're saying, right under our noses. This whole time was an amazing transitional bird, says Yale paleontologist Bharat Anjan Blair, principal investigator of this study, published in the journal Nature. It has a modern-looking brain, along with a remarkably dinosaurian jaw muscle configuration. And it's got maybe the first bird beak.
Nice.
It's kind of a proto-bird beak. It's really interesting. So it says, Buller goes on, the first beak was a horn-covered pincer tip at the end of the jaw. So it's like, it's jutting out from the top of the jaw, and it's just like, it's like a horn kind of a thing. And what they're thinking is that this sort of proto-beak was a grasping mechanism, making up for the fact that it had given up its hands for wings.
Would you give up your hands for wings?
Ooh, that's a good question. That's a tough one. I couldn't really tie it.
I'm gonna solidly say no.
But I could fly everywhere. Like I could just be like, okay, I can't type, so I won't text you back. But I'm gonna just.
I really like my opposable thumbs. But back to the seagull-y thing, seagull dino. So this is pretty interesting. Looking at the illustration on this article, the teeth look very not teeth-like, and they look a lot more like the serrations that you'll find on a duckbill. So they definitely look like this in-between state where if you look at Archaeopteryx's face, it's definitely very much a potentially a face with kind of a bill-shaped face that has teeth on it. So this is definitely a different. This looks much closer towards a bird beak. Absolutely.
Right. And these are small teeth, and it's also that they think they had not lips exactly, but they were covered. They wouldn't have been exposed.
Interesting.
Right? They would have been probably pretty hard to see if you were just looking at the creature.
Missing bird link, I see.
Yeah. And it says, this extraordinary specimen reveals the surprisingly late retention of dinosaur-like features in the skull. And it's one of the closest known relatives of modern birds from the dinosaur times.
I love this. I love when we find things in collections that are super important.
Yeah.
You're like, oh, wait, look over here in this drawer. Oh, my gosh.
What's also kind of interesting is that I think that, the brain that they're saying is very much a bird brain. It's pretty much there first, and the rest of the morphology comes, along much later. But then you can start to sort of think, well, a lot of bird brain based activity, if it pre-exists being a bird, that may give some interesting insights.
Yes, that's true. Well, I think that as we explore the fossil record, this whole archosaur branch of the tree of life that is dinosaurs and birds and crocodilians is going to become more and more blurred. So kind of like this whole idea of modern humans, and there were these all these other humanoids, and turns out, oops, they kind of like weaved in and out and in and out. I think that as we take a deeper look, as we take a magnifying glass to almost any branching on the tree of life, you're likely to see some weirdo crossovers and some potential interminglings before final breaks and all this kind of stuff. So I think this is very much in line with, as we learn more and as we find more, we're going to see these weirdo middle zones of, is this or is this that? And it's again because it's all about our lust for categorization.
And that categorization has a use. It puts things in context and allows us to learn from them and refer back to them.
But life does not live in a box. So things can mostly fit inside certain boxes, but there's always going to be weirdo things that don't belong in any of the boxes.
And one unweirdo thing about this is that they Buller's lab had already kind of zeroed in on the genes in the beak and the palate being patterned by the same genes. so that it makes perfect sense based on what we already knew, that this would be how a beak would first sort of emerge.
And I love that. I love that when they found the missing link, quote unquote, for turtles, for the origin of turtles, they found exactly what they anticipated. They found flattened ribs that were starting to protrude out of the skin. So this is, you can tell based on what you see what the middle zones should be, and it's actually really encouraging to find those. That's pretty cool. Nice.
Missing link of birds. Where they were birds.
You know what else flies?
Time?
Bats.
Also bats. Yes.
Yes. So I have a story about hoary bats. That's Hoary.
Thank you.
Hoary bats. Hoary is an adjective that means gray or white, as if with age. So hoary bats are from North America. Wake Forest University in North Carolina did a study on inconspicuous echolocation in hoary bats. What the heck does that mean? Well, they found that they were not finding bats. The way you find bats normally is you monitor echolocation. And they were all of a sudden discovering that there were bats that were being unusually quiet. So they used specialized infrared cameras and ultrasonic microphones to record bats flying through river corridors in Northern California. And they discovered that bats use short, extremely quiet microcalls. And at times, they found no echolocation at all detected, even from bats flying within 10 feet of highly sensitive audio recorders. So then they wanted to see what the heck was going on. So they stretched a mist net, which for those of you that have never collected birds or bats in the wild before, I know it might be a few of you, a mist net is a really thin net that you stretch across an area that usually is where it gets very misty, so it kind of blends in so the animals can't see the mist net. And the net, when a flighted animal flies into the net, it creates this kind of little pocket. It's a very loose net, and so the animal gets caught in the net, and then a researcher can record them or tag them or whatever, and then they can very easily open up the net and the animal can fly away. So they spread these mist nets across the natural flight corridor where they were studying these hoary bats, and they found that at first the bats used microcalls or no calls at all approaching this mist net. And when they flew into the net, only microcalls or no calls were detected on the initial approach. They began producing normal echolocation calls within a few feet of the net when it was too late to go back. So this is really interesting. This is something that we did not anticipate. Researchers have always historically monitored bats and monitored bat populations based on recording echolocation. So this is kind of earth-shaking in the bat research community. The idea that bats might not use echolocation as often as we anticipated. So they think that this actually might be happening in response to mating behavior. And so the hypothesis is that it makes them less conspicuous to competing bats and to potential mates. So they can kind of sneak up on mates, and they're not eavesdropped on. So they drew this conclusion because that was the only time of year that they saw this weird microcalls or lack of echolocation happening. But this is really important because, first of all, our population estimates are probably all out of whack if there are bats that don't use echolocation during certain times of year. But it also creates a problem with obstacles, because if we are putting up obstacles that they are not anticipating, they will get caught, they will run into things, they will get in trouble. The really big problem is wind turbines. So before any wind turbine is constructed, there's intense acoustic monitoring as a part of the environmental assessment. So that's how they figure out, is this a bat corridor? Could not hear any echolocation. Let's put up that wind turbine. But that can be a fatal mistake if there's this micro echolocation that's undetectable that could be creating these wind turbines in these corridors that actually are really beneficial, really important to bats. So the next step is now miniature bat backpacks with an audio recorder and a tracking device attached to find out exactly where and when they use echolocation and not. So if that's the case, if there's certain seasons, for example, that they don't echolocate, then you can't do your environmental assessment then, or you have to do an environmental assessment in some other way during that time of year. Or if these micro echolocations are detectable, but with different equipment, that means an adjustment in equipment. So they're a little more complicated than we anticipated.
So every once in a while, I hear what must be the bat word for man on porch, or I don't know what it is.
Yeah.
But I'll go out and there's a lamp that's out on a grassy area just outside my back porch. And pretty much every night there's bats zipping around this thing because the insects get attracted to this light, and so they're zipping around out there. And I'll go sit down out there and I can hear them every once in a while, a little chirp, and they're just flying around in circles really quick, little chirp here and there. And then I'll get out, I'll stand up, and there'll be one loud chirp, and they're all gone.
Yeah. Or it's a loud chirp going, shh, human.
Yeah. Oh, yeah. Maybe they're still there. They're just being.
Yep. He'll hear us. He'll spread our secrets.
Those giant human ears.
Yep.
It's not proportional to a bat. It's just they're bigger.
Giant, ineffective ears. Yeah. So bats, they have a lot going on, and if we're gonna save them, we need to stop again. Really, kind of typecasting those bats as echolocators. Turns out they're more complicated. Justin, you have any more stories?
Well, I don't, but Stephen Hawking does.
Oh, please do tell.
So yeah, he's got a brand new theory. His final theory on the origin of the universe. He worked on this in collaboration with Professor Thomas Hertog. It's been published recently in the Journal of High Energy Physics. The theory is based on string theory, and it predicts that the universe is finite and far simpler than many of the theories out there have been. So it's not that there isn't still a big universe and even maybe pockets of different universes, multiverse, but maybe smaller. So this guy, and I don't pretend to understand this, but modern theories of the Big Bang predict that our local universe came into existence with a brief burst of inflation. Tiny fraction of seconds after the Big Bang itself, the universe expanded at an exponential rate. Widely believed, however, that once inflation starts, there are regions where it never stops. It's thought that quantum effects can keep inflation going forever in some regions of the universe, so that globally, inflation is eternal.
Inflation is eternal.
Yeah, but but maybe not. So this is Hawking in an interview last year. The usual theory of eternal inflation predicts that globally our universe is like an infinite fractal with a mosaic of different pockets, universes separated by an inflating ocean. Local laws of physics and chemistry can differ from one pocket universe to another, which together would form a universe. I have never been a fan of the multiverse. If the scale of different universes in the multiverse is large or infinite, the theory can't be tested. So
what's the problem? Can't be tested.
Yeah.
I mean, I can't know for a fact that there's a universe where we're all wearing cowboy hats right now, but it might exist.
Well, in the paper, Hawking and Hertog say this account of eternal inflation as a theory of the Big Bang is wrong. problem with the usual account of eternal inflation is that it assumes an existing background universe that evolves according to Einstein's theory of general relativity and treats the quantum effects as small fluctuations around this. However, says Hertog, the dynamics of eternal inflation wipes out the separation between classical and quantum physics. As a consequence, Einstein's theory breaks down in eternal inflation, which would be, I think, just about the first time we've heard of an Einsteinian theory breaking down under anything. We predict that our universe, not theirs specifically, but just the one we share, on the largest scales is reasonably smooth and globally finite. So it's not a fractal structure, which I didn't think it was either.
So does that mean eventually it's going to slow down? It's going to slow to a stop is the theory then. If it's not, I
don't know that it says that. I don't know.
Well, that's the opposite of eternal inflation, right? Is non-eternal inflation. So it's going to stop.
You're right. But this is also about different pockets of this taking place differently in different places. Not our universe, but another universe outside of our universe that might be expanding entirely differently. They developed a variation of the holography concept. This is a Leonard Susskind kind of thing in string theory. To project out the time dimension in eternal inflation. This is the idea that physical reality in certain 3D spaces can be mathematically reduced to 2D projections on a surface. This gets into kind of like we're living into the Matrix. Once you start overanalyzing how physics works. It can be scary.
Well, so, and talking about time scales that are not even fathomable is hard. But I'm trying to think about what they're saying, and I'm probably totally misinterpreting this, but I'm kind of picturing a melting stick of butter in an empty Olympic-sized swimming pool.
Right. Say this again.
A melting stick of butter in an empty Olympic-sized swimming pool, because it's a finite space, but it doesn't really matter.
But wouldn't it be totally viewable by this surface? Because it would be all the lipids would be.
No, empty. An empty swimming pool.
Oh, it's empty. There's no water.
It's empty.
There was nobody else in there.
No, there's no water in it. It's just that's the container.
Then why is it a swimming pool?
And you're melting butter. So
is it a hot day? It's summer. How come there's no water in the pool?
It feels endless. The space that this butter could expand out into, but it's not. That's what it's feeling like to me, but I could be way oversimplifying it. I don't know.
So there's a potential to test their theory with
what now?
With LIGO detectors. Hertog believes that primordial gravitational waves, ripples in space-time, generated at the exit from eternal inflation, constitute the most promising smoking gun to test the model. Don't know what that means, but we may find out one day whether or not the theory is correct. Space may make some people feel insignificant in its size and scope, but not me. And I was actually heartwarmed by my last quick story. And this isn't there's a pair of researchers that took up the task to figure out the longest route you could sail by sea in a straight line in the oceans, right? What is that? How far could you go? When they got out their ruler, though, to figure this out, it turns out the world is round. When the ruler was applied to the globe to plot the course, it plotted the course out into space. So not very long if you're going to keep a straight line.
My brain, not ready. Not ready.
Well, the real story is they came up with two courses. And I guess because of the way the Earth goes, I don't understand. But apparently you can go a really long ways. 32,000 kilometers. Is that right? Yeah. 32,000 kilometers in a straight line if you pick the right route in the sea. You leave from Russia and somehow you end up in Pakistan. But as I look at this, I get that the world's curved. I just don't see how it's curved that way. That's like the way a satellite goes.
Yeah.
No, I say it's a ruler. You end up out into space and you can't go in a straight line anywhere on Earth or very far.
Yeah. Well, I'm glad to hear research from Stephen Hawking making its way. I like to imagine him and Einstein in an argument in a laboratory in heaven if it existed. It would be fun to see and hear. I wouldn't understand any of it, but it would be neat.
Yeah. And then somebody goes, "Don't you know all of the answers have been revealed as soon as you get here?"
Oh yeah.
Open up the book.
They're like, "But what's the fun in that?" That's no fun. That's not heaven. Okay.
Einstein and Hawking, the dumbest people in heaven.
I have a fun story, nice and quick one about the world's oldest spider. World's oldest spider. Where was it discovered, Justin?
How is it a big spider?
It's a big one.
Then it's in Australia.
It's certainly in Australia. Ding, 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 ding. This is a trapdoor spider, one of the largest types of spiders, and it has been found to be how old?
So you're asking me, and I can tell it's a trick question because I'm going to say something along the lines of many, many millions of years.
No,
you're talking about like not how long has it existed, like
right. This individual, they found a matriarch Spidey in Australia. This Spidey in Australia is the oldest spider they've ever found. Wow. How old?
So I have no idea how old a spider can be, but I'm going to say something in the neighborhood of like 3 years.
Okay, good guess. A little bit more. So previous record holder was a tarantula in Mexico at a ripe old age of 28.
Oh, wow.
Years old.
Wow.
This spider blew him out of the water. This spider was discovered by Barbara York Main in 1974 and has been monitored since. The spider died just a couple of weeks ago at the age of at least 43.
Oh, right. Because when they found the spider, it was already spidering around. I don't know.
Already spidering.
The age of the spider when they found it. So I don't then be able to tell exactly the age, but at least 43. That is 40 years more spider than I would have guessed, not having known how old a spider can be. What is that? What's a typical spider age? Like
a few years.
Okay, so I wasn't like completely off the scale of like, most spiders make it till
it depends on it depends. I mean, tarantulas usually live pet tarantulas live 5 to 10 years. That's pretty normal. But so the thing about this study that's really amazing, first of all, people don't know spiders can live to be that long. We didn't know even the current record holder at 28 years old. This was 43 years old. That's way older than that. But what's really interesting about this study, if you ask me, is that Barbara, who started the study in 1974, she started recording this individual matriarch. This was at Curtin University. Her lab continued her research after she retired. She's still alive. She's 88 years old. And they have continued her research, and they have continued watching this matriarch. So they were able to definitively say that this spider at 43 at death was the oldest spider ever recorded. So they were able to record age, cause of death, and all this kind of stuff. They were able to record life history. So this demonstrates a really sweet thing that lab did, but also it shows you why long-term research is important. And with a lot of the way academia is set up, not a lot of research gets carried on, not just from graduate student to graduate student, but after professors or researchers retire. So I think that this is a good reminder of why it's so important, especially when we're learning about life strategies and about animals that we don't know a lot about, that continuing these long-term studies can be super important.
Although I'm already thinking that somewhere, 20 years ago, some lab assistant made a mistake.
Counted the wrong spider.
Happened. We're supposed to just clean the thing. I didn't, wasn't paying attention. Luckily, I got a friend that works at a pet store, ran over, grabbed another spider, put it in. They never knew.
This is a wild spider. This is not a lab spider. Which makes it even more amazing.
What do you mean it's a wild? Wait a second.
Yeah.
What do you mean it's a wild spider?
Yeah, they were
able. Not in the lab?
No.
Then how do they, they're not watching the spider?
No, they know. They've identified markings. Maybe they've marked the spider. They know. These are spider people. They know this is the same spider without a doubt. That's like saying, how do you know that's your tabby cat?
No,
They know. They know that's the right spider. But absolutely.
Well, that's a lot of, I don't know.
That's the way these things work.
Well, okay, why did the spider die? Did a bird eat it?
Why did the spider cross the road? No, I don't know. Actually, they haven't released cause of death, but they were able to record it, and it sounds like it was probably natural causes. Because they're saying that this is what they think might be a representative lifespan. Wow,
that's pretty impressive.
Yeah, absolutely. Do you have a last story, Justin?
That was it.
You're all done. I have one more story. I thought you had another story. I thought you had, what's this, plotting the course?
That was the one about the guys who were trying to figure out how far you could sail.
Right. Yes.
I didn't really talk about it.
All right. Okay. Fair enough. Okay. So I have one more story about horses. So horses are pretty smart. We've talked about that on the show before. They've been with humans for a very long time. But a new study from universities of Sussex and Portsmouth found that horses can read and remember human emotions. In this study, they took domestic horses. They presented them with a photograph of an angry or happy human face, waited several hours, and then had the actual person from the photographs walk in, sit down, and exhibit an emotionally neutral state. They found that the short-term exposure of the photograph of a person's facial expression was enough to create a difference in response upon meeting that person in the flesh. So despite them having a very neutral facial expression in the live meeting, they saw a difference in horse gaze. And this is something that has been studied before, is that animals, and in particular horses, tend to view negative events with their left eye due to right brain hemisphere specialization for processing threatening stimuli. So left eye, right hemisphere. And so they were able to watch that people, so the pictures were shown where they either were smiling, really happy, or they were shown a picture where they looked like this. All scrunched, frowny.
That's not a happy,
squinty face.
Ape face. No. Oh,
yeah.
No.
So they showed them these pictures. And then they had them come in, sit down, neutral expression. Nothing special in the eyes, nothing special in the mouth, just neutral expression. And they found that the people who had the scrunchy, mean face in the picture, those horses, left eye gaze. So they also made sure that in the experiment, humans didn't know which photographs the horses had seen before they came in. So they weren't expecting anything. They also saw that the differences in reaction were only applied to the person the horses had actually seen in the photograph. So if they brought in somebody who they hadn't seen in a photograph, they didn't do the left eye stare. So Professor Carin from University of Sussex says, what we found is that horses can not only read human facial expressions, but they can also remember a person's previous emotional state when they meet them later that day. And crucially, that they adapt their behavior accordingly. Essentially, horses have a memory for emotion. This is the first time that it's been shown that horses can remember emotional experiences with specific individuals. There you go. Horses, they know what's up. And as somebody says in the chat room in our YouTube chat, horses have been with us for a long time. I wonder if the same thing would happen with wild horses. I would guess no. I would guess this is absolutely because these animals are, for tens of thousands of years at least, tied to us, and they have evolved alongside us. They don't look like horses looked before we brought them in with us. So I would guess that if you did this experiment on dogs, you'd see similar results. But if you did this experiment on a wolf or on a zebra, you'd see nothing.
Wait a second, though.
That would be my guess.
Right, but I think that there was a study along these lines, and the wolf did better than the dog.
So that was, you're right, but that was,
and
there
sort
was
of telling
some,
like, okay,
but
like,
they weren't. So
tweet
that's
under
the
one
difference.
cup. I'm gonna look at one to indicate which it was. The dogs were slower to pick it up than a wolf.
Yes.
Was
So this was in the instance where the face was threatening or not. They weren't asked to recall it with specific individuals. So that's where this is different, is that they are remembering individual faces and the context that those bring.
And can't octopi do that?
So octopuses.
Octopi, that's not a thing.
Octopuses. That's a good question. I don't remember a study about human emotion with octopuses.
No, not necessarily. I guess not emotion, but they, for whatever reason, would, and maybe it was because they felt like they got stink-eyed or mistreated. They could, if they don't like you as somebody who's working in the lab or the aquarium, and you leave and are gone for a while and you come back, they remember and might like spit water at you.
Yeah.
Or...
So octopuses and other cephalopods are really good at associative memory. So they'll remember stripes mean squid and squares mean crab. So I'm not surprised to hear that they can learn about specific individuals. That makes a lot of sense. But I think that what is so particular about this study, and I haven't seen replicated before, and hopefully we'll see more of it now, is the combination of face recognition with context memory. So the idea that it's not that they got any actual negative stimuli as a result. It's just that you saw a sour face from a picture of a person, and then that individual showed up in real life and you were wary of them. So that's something that even with crows aren't predisposed to understand human facial expression, but they are predisposed to remember context and pass along context clues.
The reason I think the wild horses might still be able to do this, though, although you'd have the whole problem of them being uncomfortable maybe around people, so it would be like impossible to do this study maybe. But they use facial, physical posturing, ear.
The ears are big,
They use a ton of visual emotive cues amongst each other that may be pretty easy to translate into a human face as well. It's what we're good at, and we can probably recognize pretty readily how a horse feels. If you're somebody who has horses, you're pretty comfortable with the mood your horse is in, right? So I would expect that ability would be there, but they would probably be just more concerned about, a wild horse would be just more concerned about getting away from it.
Right. So then you could try to replicate this study, but I think it would have different implications. But you could, for example, take pictures of other horses that are stressed out versus comfortable, show them those pictures,
and have the horse come by now totally calm.
That horse. Yes.
And see,
and see what happens if they get uneasy when the horse that they saw is stressed out in a picture comes by. And so there's other extrapolations of this. But I think what's important about this study is that they were attuned to human faces, and that then they remembered that when they met the human in person. And that's the combination of all these things that we've tested in other animals and other studies. But the combination of all these things together says, to me at least, that these animals have evolved alongside us and have been bred alongside us, and they are very well attuned to that. It is important, it is advantageous for them to be aware of and store information on human temperament.
And I would still disagree a little bit in saying that I think that's also just one of the natural skill sets of a horse. I don't think your cat would care, regardless of how long cats have been domesticated. I bet you can show
cats. I would argue are not domesticated.
That's okay. That's a good. I would kind of agree with that too.
Yeah, they're not.
But they've
cat
been
can
with
survive
humans
without...
for so long, and I bet you if you did try to do the same study with cats, they would just look like they didn't care no matter what.
Yeah. So I mean, cats are a weirdo thing because we've kept a lot of bizarre wild traits in these animals that we have in our homes. So it's a whole extra mess of weird variables. But with the horses have been a tool that humans have used for tens of thousands of years at least. So
and I think though that possibly part of how that relationship began and got started and why they I don't know, maybe you're right. I was about to say there's a reason that we're not out on the plains riding cows, but also slower.
And there's a reason we're not out riding zebras either, but
can't people ride zebras? Nobody does that. There's none.
No, you don't want to mess with a zebra.
They're like mean donkeys.
Yes.
Yeah.
Yes. They'll sneak up on you. Anyway, I think we've done it.
I think so.
I think that's the show. All right. Well, we are going to start our wrap up tonight with a shout out to everyone on Patreon. I do not have access to the list because the Grand Master, Dr. Kiki, has access to that login information. But I will say a huge, huge thank you to all of our Patreon sponsors, but especially those that are in particular interested in getting a shout out on a show. This is your shout out for this week. We appreciate you. Shout back at us. We'd love to hear from you. If you are interested in supporting us, again, you can check us out at patreon.com/thisweekinscience. And you can also help us out just by spreading the word about TWIS. On next week's show, Dr. Kiki will be back. Huzzah! And we will be broadcasting live at 8 p.m. Pacific time on twis.org/live, where you can watch and join our chat room. But don't worry if you can't make it. You can find our past episodes at youtube.com/thisweekinscience or twis.org. And a quick thank you to everyone who helps get us to all the places that we exist. I think it's Brandon helps us get on Facebook. Fada helps us with our show notes. I-94 helps us with our sound. Very important. So, and everyone else who I don't have written down because I am not Kiki and I am not as great as her, know that we care about you, we love you, and we thank you.
Yes. Thank you for enjoying the show. TWIS is available, of course, as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science and anything Apple marketplacey.
For more information on anything you may have heard here today, show notes are available on our website. In case you haven't heard, that's www.twis.org. That's www.twis.org. While you're there, you can also make comments and start conversations with the hosts. Hey, that's us. Or other listeners. Hey, that's you.
Wait, I was trying to write down that website. What was it? You said it so fast.
Oh, it's TWIS.org.
Okay, it's fine. I found my place.
Okay,
or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair Baz at twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filled into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here, whole, all 3 of us, next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
We did it. We did a show. Those of you on YouTube, I was scrolling through some of the comments and I noticed some questions about classification. So I'm just going to real quick address. So the platypus is an egg-laying mammal. The only other mammal that lays eggs is called the echidna, spiny echidna. So those are the two. And then there was also a question about red pandas. Okay, so red pandas are mammals. Red pandas are carnivores. They are in Carnivora. They used to be classified with raccoons, but they are no longer classified with raccoons. They're all by themselves. So red pandas. There were some questions in the YouTube chat about that. There were also questions about platypuses. So mammals, the thing that identifies all mammals no matter what are, Justin?
For the most part, live birth.
Not 100% though, because you have the platypus and the echidna that lay eggs. So think about the word mammal. Where did that word come
mammary.
from? Mammaries. They all have mammaries. They all make milk. Even the platypus and the echidna, which do not have nipples, still create milk. Lactose-based sustenance for their babies. It's just kind of like it sweats out on their tummy, and it goes down. They have specialized hairs that it just flows down these specialized hairs, and the baby just kind of like lick it off. The specialized hairs. So I did a report in college on the type of lactose in platypus milk. It is a special protein only found in monotreme milk. So monotremes are egg-laying mammals.
Wow, where can I get some? That would be like
good question. I'm guessing you won't like it. But yeah, then they were asking at red pandas. Red pandas are normal mammals. They have live birth. They have everything that other mammals have. They are they're in carnivora, so they're related to bears and wolves and cats and all these things. But they used to be classified in the same family as raccoons, but they are no longer in the same family as raccoons. They are in their own family. There you go. There you go. Fun fact. Hashtag fun fact.
And they have 40-something gut microbes in common with panda bears.
Yes, because of the bamboo.
And the bamboo-eating lemur.
Absolutely, bamboo lemur. And Dave Shorty said eggs and milk from one creature. It makes me want to farm them. So the eggs that come out of platypuses are like the size of a Hershey's egg you'd get for Easter. They're like this big. They're teeny tiny. So they're funny. But so the reason that they still lay eggs is that they're the blastopore, the rudimentary thing that isn't even a fetus yet, that is a developing ball of cells, like the pre-fetus, basically. In us, it has these huge, what are called invaginations. So they're basically like wrinkles all around it so that it can implant into the uterine wall in mom, and it allows for gas and nutrient exchange until the development of the placenta. So the placental mammals are what we consider like normal mammals because they have this placenta, which allows them to cook in mom for a very, very, very long time because they have this gas and nutrient exchange that is extremely fancy and well developed. On the opposite end, you have monotremes. You have platypuses and echidnas that they're like a ping pong ball. They're like very smooth. So they have trouble kind of like attaching to the uterine wall of mom. The gas exchange is not great. And so they have to be plopped out sooner. And so that egg protects them when they're still very small, when they're not even really a fetus yet. So it allows them to pop out and they're contained in these eggs. The in-between between those two are marsupials. So marsupials, they have some invaginations. They have some of this wrinkliness on the ball of cells so that they have some gas exchange with mom, but it is not as developed and there is no placenta. And so as a result, they're born very early when they're the size of a jelly bean and they're still blind and their back legs don't work and they have to crawl by themselves into their mom's pouch. And then they stay in that pouch until they're big enough to be able to go out on their own. And fun fact about that is that the mom actually has 2 different types of milk and 2 different types of nipples in her pouch. So she has a high protein and a high fat milk. So the high protein fat is for the teeny tiny little thing that's still developing all the important stuff. And then the high fat milk is for when they're bigger and they're coming in and out of the pouch and they're just kind of bulking up. So
and for those who may not also know, describe koala pap.
Oh, it's just basically poop. It's poop that is infused with the bacteria that allows them to digest eucalyptus. And so baby koalas. So here's another fun fact. Koala pouches upside down. So the baby koalas are poking out towards the bottom of the koala The mama
Seems
koala.
like an inefficient.
So no, it's perfect because they just poke out, eat the little pap, and go back in. So they have their bacterial seed that way.
So the a baby koala's first meal, because before they
they're drinking milk. They're drinking milk.
They can take milk, but before they can even digest eucalyptus, they need
to eat
a
poop.
fecal transplant.
Yep, sure do.
Wow.
Yep. I'm full of these. In case you were curious. And yes, the plural of platypus is
platypuses. Platypi?
That's a great question. I'm gonna look it up actually. I always get it up. So octopuses.
Wait, where are you getting some of these questions? I'm not seeing all of these.
I'm in YouTube now. Kiki usually does that.
Oh, really?
So yes, platypuses is correct. So platypuses. Platypi is incorrect. So the problem is it's Greek. So octopus and platypus is Greek. So you would say platypuses, or if you're being super correct about the Greek, you would say platypodes, which is why you'll hear people say octopodes occasionally. But really, because we're an English language and we are pluralizing based on English rules, you are supposed to say platypuses or octopuses.
I thought it was platypods. And
no, it's pod.
Pod.
Octopods.
Octopodes.
That's why I'm here. I'm here to educate. That's the science we're learning. I'm so happy to clear it up. Oh my goodness. All right. What do you think, Justin? You have anything you're dying to talk about?
Yeah, no, I can't stop thinking about the quid spitters.
Yeah.
And I realize I now have access to genetic sequencing equipment, which means I could go around town and
just pick up gum?
Pick up bubble gum from the sidewalk, pluck it off from underneath tables at the diner, and I could start tracking down people and be like, excuse me, I think you dropped this. Just throw it at them and leave.
Well, pretty soon you'll be able to do DNA tracking on like vandals and things like that, huh?
If they spit out their gum, I guess.
Yeah.
What did you get vandalized? What happened?
No, I'm just saying. Be pretty funny.
So we got I got to play for the first time with the nanopore thing.
That's fun.
On it, and the software is called Minion, which I thought was really awesome, but nobody else there understood why I was so excited that it was called Minion.
Yeah. Well, it's whenever the Despicable Me movies come out or the Minion movies come out, it's kind of fun. Just because you see the word minions everywhere, and you're like, that was us first. We had it first.
Yeah. But I guess we did it. We did the show. We did some after show. You took round robins of questions. We could call it good. We could take off.
My throat. I'm trying to keep things from happening. So it's been a rough few weeks.
Fighting off the cold. This is what you're hinting at.
Yeah.
Okay.
I'm trying to make sure I still have a voice tomorrow. I have to teach high school students tomorrow about climate change, which I did today, and I'm going to do it again tomorrow. And I'm so excited about it. I love doing it. I love looking at their dead faces. They're just like, but then at the end, at least some of them, it's clear that they're excited that you came. They couldn't show it during the class. It's always when I walk around with my stickers. Everyone's like, stickers are dumb. And then I walk around and they're like, I'll take one. Like, yeah, got him. Pew pew. Got you with the animals.
I'm gonna share this, but this is something. We had a congressperson visit our workplace today.
Ooh, that's cool.
There's all these like ridiculous like emails that I shouldn't be talking about work, but I'm going to a little bit, that were like, we want everybody to like be busy doing something sciencey. It's like it's what everybody's always doing anyway. But it was sort of like funny, like
well, it's like when the boss walks through and I'm like writing a Facebook post, and so I'm on Facebook because that's my job, and then I suddenly feel really self-conscious about it. Right. Like, oh no, they're gonna think I'm messing about here, and I feel like I have to like put up a sign that's like, no, really, I'm working.
Yeah, and I think there's something to it too, maybe there, because I think I kind of just like assume that everybody there is my boss because like they're all, have been doing what they're doing for forever or longer. And so maybe I'm already kind of on the point of like always trying to make sure I'm keeping busy, that I don't notice if other people aren't normally here. But I don't know. It seemed kind of funny because it's a pretty busy place to then like.
Yeah. That's pretty fun.
Yeah, that was fun. It was exciting. All right, I'm out.
Okay.
Good night, minions. Say goodnight, Blair.
Good night, Blair. Say goodnight, Justin.
Good night, Justin.
Good night, minions. Bye.